Hi, and thanks for having me here today. I'm going to talk about the relationship between spontaneous brain activity and specifically Hertz exponent as a proxy for long-range temporal correlation and glucose metabolism. So basically, I'm going to combine dynamic FDG PET and kinetic modeling of dynamic FDG PET and resting state fMRI. Now, as you can see from this figure, the brain is an energetic outlier because it accounts for just 2% of body mass, as you can see here, but it consumes about 20% of body total energy. Here you can see this paradox occurring in a real-life scenario of an FDG PET scan with the brain being the brightest organ. If we zoom in, this time using the same tracer, so always FDG, but with a dynamic acquisition, I register to MNI space. You can even see how heterogeneous is tracer uptake, not only over time, but also over space, with specific areas like deep precuneus, for example, having much more radioactivity as compared to other areas, for example, motor cortex. Even more paradoxical than the size-to-energy ratio I told you about in the previous slide is that most of this energy is being consumed at rest, such that performing specific tasks like providing motor responses, perceiving visual stimuli, like reading sentences, only increase local energy use by up to 5%, so very little. And that's the so-called brain dark energy. So most of this is being consumed at rest. Now, David Atwell has done very interesting calculation showing that most of this is being consumed for synaptic transmission, as you can see here, and not for action potentials. And even if we take housekeeping functions into account, still the vast majority, as you can see here, is being used for information transfer, which draws a strong link between spontaneous activity and glucose metabolism. Now, one of the most striking features of spontaneous activity are long-range temporal correlation that basically reflect the scale-free architecture of brain functional activity, and that they have been observed, as you can see here, across many modalities, from EEG to calcium imaging and resting state fMRI. Now, there are at least two reasons why they could be related to glucose metabolism. Now, the first line of evidence is related to the fact that they fall together in states that are pushing the brain away from criticality, like loss of consciousness. For example, here you can see a study by Enzo Tagliazucchi. And as you can see, the Hertz exponent is dropping when we move from wakefulness to propofol-induced sedation and then loss of consciousness. And then they rebound when consciousness rebounds. Now, the same exact pattern occurs with glucose metabolism because we have a massive drop plus a regional pattern that it's very similar to the original drop of Hertz exponent in loss of consciousness. Now, the second line of evidence instead refers to asymmetries in both information transfer atop the structural connectome as well as in terms of glucose metabolism, because several studies have shown that hubs of the human brain operate at slower timescales as compared to peripheral areas. And in the mouse brain, in-degree hubs have shown slower timescales as compared to out-degree hubs. And the same asymmetry occurs with glucose because Atwell has shown that most of glucose consumption happens at the level of the postsynaptic neuron, which, if we scale this at the system level, neuroscience indicates that in-degree hubs should have both higher glucose metabolism as well as lower timescales and stronger long-range temporal correlation. Now, based on these premises and theoretical background, we made some prediction we wanted to test on a multimodal dataset comprising dynamic FDG PET. It was a bolus protocol that's ideal for kinetic modeling, as well as resting state fMRI, that they were acquired sequentially using two different scanners, but all from the same set of subjects. Now, we wanted to test two predictions. Firstly, that the Hertz exponent would track regional glucose metabolism, and then an across-subjects relationship, meaning that individuals with stronger scale-free activity should also display a higher glucose metabolism if these two are related. Now, we used a popular wavelet-based estimator for the Hertz exponent, which is very popular, again, nowadays. And we also measure how reliable it was across different sessions, since we had access to 2 sessions of resting state fMRI with opposite phase encoding. And we found that it's pretty reliable, as you can see from the second plot here. As for the PET data, we wanted to distill as much information as possible. So instead of using simpler semi-quantitative or graphical methods, we did full kinetic modeling of FDG, specifically the two-compartment Sokolov model that allows you to get not only the KI, which is the net tracer uptake and can be seen as the CMR glucose in many papers, but also microparameters that provide a full description of the first step of glucose metabolism. Specifically, we had access to K1, which is measuring basically the influx of the tracer across the blood-brain barrier, K2, which is clearance of the tracer into venous blood, and then K3, which is the enzymatic activity of the hexokinase enzyme, which is the actual glucose consumption. Here you can see 3 maps. We have the KI, so tracer getting in, tracer being phosphorylated, and then the net tracer uptake. Here you can see the maps once we apply the parcellation atlas, so you can even appreciate how heterogeneous they are over space. For example, the K3 is more unimodal, transmodal, as you can see here, as compared to the KI, which is again closer to the CMR glucose. Here you can see 2 example subjects. So in the end, in our data set, we had access to 5 different maps. One related to the Hurst exponent based on fMRI, tracer getting in, getting out, being phosphorylated, and overall uptake. As you can see, there are differences not only across brain areas, but also across different subjects. Now, the first thing we did was to associate these measures at the group level. So we are basically measuring correlation between Hurst exponent and each of the glucose parameter we extracted. And we found a strong positive association with KI. So where in the brain the Hurst is higher, also the uptake of the tracer is higher, as well as with the K3. So there's a faster enzymatic activity of the hexokinase enzyme, where in the brain long-range correlation are stronger. We also replicated these patterns at the single subject level. So each dot here is a correlation coefficient. And we are basically seeing the same results I showed you in the previous slides, but at the single subject level. So these are again spatial association. We then analyzed across subjects relationship. So we did regress the average Hurst exponent map, and we did try both with and without global signal regression onto the glucose microparameter, while controlling for age, sex, and overall in-scanner motion, and found a strong positive association with the K3, which indicates that individuals or subjects with stronger long-range temporal correlation have faster, on average, enzymatic activity of the hexokinase enzyme, which is being phosphorylating the tracer. As a very last step, since glucose metabolism is both a process itself as well as a signature of other processes taking place, for example, synaptic plasticity, we wanted to add more biological information to the model. So we added 2 more tracers based on independent data, one targeting synaptic density, and the other the in vivo de novo rate of protein synthesis, which is this one here. So we developed a very simple multiple regression model in which we regress the average Hurst exponent as a weighted linear combination of glucose metabolism. Here we use the KI, synaptic density, and protein synthesis. And we also did some kind of dominance analysis to rank predictors depending on importance, and found that rate of protein synthesis, so a dynamic activity, was the best predictor of the Hurst exponent, as you can see here, while glucose metabolism was the second best. Synaptic density did a very poor job in explaining the regional pattern of Hurst exponent. We also did some kind of cross-validation inspired by this paper by Hansen. Now, jumping to the conclusion, we basically found that the scale-free architecture of brain activity has a significant energetic cost that we investigated not only across space, but also across subjects. And it seems that the ability to generate persistent activity may depend on very fast dynamics, including a faster enzymatic activity of the hexokinase enzyme, as well as faster biosynthetic processes. As for future developments, I can foresee this coupling being investigated, for example, during task-related activity, because the vast majority of ATP production at rest is due to oxidative phosphorylation, but a small fraction is produced by aerobic glycolysis, which is way less efficient, but could be faster, and therefore could be important in task evoke activity. So I want to thank my PI and collaborator, and thanks for your attention.